Okay, thank you for the introduction. As was mentioned, my name is Meaghan Parks. I'm a graduate student in the Cancer Evolution Lab at Case Western Reserve University, and I'll be talking about uncovering cancer's fitness landscape. So in recent years, there have been numerous advancements in synthetic biology. Technologies such as DNA microarrays allow us to measure gene expression on thousands of genes simultaneously. And advancements in gene editing have allowed us to control gene expression or even just completely knock out or just limit gene expression. So the combination of these and other technological advances have led to multiplex approaches, which have in turn led to an unprecedented amount of genetic information. A very good example of this is the Cancer Dependency Map, or DEP-map, which is a broad consortium-led effort to determine all cancer-dependent genes. Now, as you can see here, this is a data set, it's just one of their data sets, and it contains 1,000 cell lines in which they knocked out, in every single one, 17,000 genes. So it's a massive amount of data. Now, when we're talking about mutation types today, I'm going to be talking about adaptive mutations that increase an organism's fitness, deleterious mutations that decrease an organism's fitness, and neutral mutations which don't change an organism's fitness. Now, this is a graph showing the effect of knocking out TP53 in several different cell lines from that same DEP-map data set that I showed you. And you'll notice here that in this data, there are, in certain cell lines, this is an adaptive mutation when TP53 is knocked out. In others, it's deleterious though. So this is an example of a behavior called sign epistasis, when a mutation and its phenotypic effects change sign based on a genetic background. And this behavior is not exclusive to TP53. In fact, it's found in numerous different tumor suppressing genes. Another example is RB1, which, as you can see here, shows similar behavior, with it being adaptive in some cases and deleterious in others. So this direct relationship between a gene being knocked out and its fitness effect is an example of the relationship between genotype and fitness. Other work has been done on this DEP-map dataset to look at the relationship between genotype and phenotype. In 2022, Pan et al. Performed dictionary learning on the DEP-map data, and through this, they were able to infer gene function. So from this one data set, we've been able to look at the relationship between genotype and fitness and genotype and phenotype. However, we've yet to actually look at the overall relationship between genotype, phenotype, and fitness. How can we do this? Well, one way of doing this is using fitness landscapes. So fitness landscapes were introduced in 1932 by Sewall Wright. He introduced the idea of studying evolution by visualizing a landscape based on fitness, where peaks are high fitness and valleys are low fitness. So fitness landscapes show us how fit a mutation is in a specific genetic background. Now, this landscape that I'm showing here is fairly complex. However, we can simplify this because complex trait theory tells us that over short time scales, there will be an optimal fitness regards to the overall landscape space. So looking at an actual timescale that we're likely to be interested in, we can essentially zoom in on one of these peaks and we get a shape much more similar to this. Now, you should note that, as I've mentioned before, sign epistasis is found in numerous cancer genes. So sign epistasis on fitness landscapes is typically found near a peak because a mutation can push you over the peak, thus lowering your fitness, or push you slightly up, increasing your fitness. So there was a model developed by Fisher and Orr also in the 1930s that treats organisms as a collection of independent phenotypic traits that make up axes in a theoretical space under stabilizing selection, meaning that there is an optimal fitness. This model was used very successfully in microbes to predict gene deletions. However, the experiments that were done were using microbes that had a single genetic background. So they were very carefully curated to make sure that they were all coming from the same starting place. So looking at a flattened landscape, this is the same landscape where the star represents the peak, just a flattened view of it. If we pick a random point on the landscape, a mutation can have three effects. It can move you further up the peak, thus being adaptive. It can be neutral, which is represented by a circle around the peak. It doesn't actually change your elevation. Or it can be deleterious, where it moves you further away from the peak. Looking at this on a completely flattened view, again, any adaptive mutation will increase your elevation, neutral will not change it, and deleterious will lower it. Now, back to our flattened view. As I mentioned before, previous work with this has been done from a single genetic background. With the DepMap data that we're hoping to use, there are over 1,000 because they're all different cell lines. They're all starting in different locations. So what we essentially have are snapshots on the landscape, 1 point Z. We have a fitness measurement for it. However, that Z can actually be broken down into 2 parts. We can consider it big Z, the ancestral phenotype, so the starting location of the cell line before it's mutated, and P, the effect of the mutation, so how it's moved on the landscape. So our endpoint, the actual fitness that we're getting, is Z plus P. Now, because of this, and given enough data, we can actually determine the overall landscape. So if we have the fitness for 3 points on the landscape, we'll know which of these points actually share an ancestor. So we have 1,000 cell lines, but we have 17,000 genes knocked out in each of them. So some of the points are going to share an ancestor. And given the fact that we know that, and given that we know that the mutational effect will always have the same direction and magnitude, regardless of whether or not which cell line it's applied to. So it will not have the same fitness effect, but it will have the same direction and magnitude. So if you look at the pair of Z2s here, P1 is actually giving a deleterious fitness effect. However, when you look at the pair of Z1s, that's a adaptive fitness effect. So it's the same direction and magnitude, different effect because it's moving you further from the peak. So with sufficient information, we can do this. We have sufficient information given the DepMap data because we have 17,000 times 1,000 snapshots on the landscape. So in theory, we should be able to work backwards and triangulate our landscape shape. So can we infer the genetic background of cancer using massively parallel perturbation data alone? So far, it seems yes that we can. So after modifying Orr and Fisher's equation, I've been able to predict the genetic background on synthetic data extremely successfully. This is the modified equation here. So that's what this looks like on a very simplified, just 2D landscape. We have the simulated fitness, which is the blue triangles, and we have the regressed fitness. So we're able to get back to our simulated fitness using the model. However, we did notice something interesting, and that's that regression fails to find a single solution. So this is a graph showing Z values. So the ancestral phenotypes that we got back. So we have our simulated ancestral phenotype, and we have our regressed. And now this particular pair matches well. However, looking further, you can notice that this is not the case for all of them. In fact, it's not even the case for most of them. In general, the regressed Z's don't match with our simulated ones. We actually gave the model despite the fitness matching. And this trend continues with P. So what we found was that multiple runs of the model resulted in numerous solutions with equivalently good fits, but very different actual landscapes. So how is this happening? Well, if we look at a 2D landscape here, we have 2 points here labeled Z. Now these have equivalent fitnesses because they have the same elevation. So there's already 2 points on this landscape that would give us the same results. However, additionally, Z is made up of 2 parts, Z1, as shown here, and P1. The combination of them gets us to that fitness. However, we can also have a combination of Z2 and P2, which gets us to the same fitness. So in other words, these are equivalent. And when we look at our regressed and simulated data, when we have Z plus P, you can see here that the Z's and P's plus P's, they share the same color on this graph. You can see that they're essentially orbiting the origin. So they're in equivalent fitness, they're going to give us the same fitness result, but they're not the same. So, and as the dimensionality of the space increases, these symmetries only become more complex. Now, the first thing we had to do was actually make sure that our regressed landscape was actually a transformation of our simulated one. So to do this, we performed 2 different types of Procrustes analysis. Firstly, we performed orthogonal Procrustes analysis, which only allows transformations of like rotations and reflections. And that performed very poorly. However, when we perform more general Procrustes analysis, which includes more types of transformations such as scaling, this performed extremely well. So this tells us that all of our learned landscapes that we were getting are complex transformations of the simulated ones. So how can we ensure a unique solution? Well, we can do this if we consider a fitness landscape described by two sets of d-dimensional vectors. So we have our cell line vectors that are joined into a matrix, and we have our mutation vectors, so our mutation effects. We can ensure we find a unique solution using two methods. So both of these methods are applied to the P matrix. In the work that we've done, there isn't a reason it couldn't be applied to Z instead. But we can control the rotation and scale by setting the upper triangle of the matrix to be 0. And we can prevent reflections by setting the sign of the off diagonal. So we're preventing it from rotating in space, and we're preventing it from growing and shrinking. And we're also preventing it from reflecting. Now, this is a somewhat biased view of what this constraint looks like, because in reality, the actual number of rows in the matrix is generally substantially larger than the number of dimensions. So while here it looks like this is a lot of changes to the matrix and it's a lot of control, in reality it's not. It's very little. So this is another graph showing P, and this is with the constraints. So as you can see here, they align up nicely, whereas before they were quite disparate. So we know that our constraints are leading us to one single good solution. Similarly with Z, we see the same behavior. And when we plot Z plus P, instead of simply orbiting around the origin, they're actually overlapping now. And on a slightly expanded data set where we have more mutations applied to the cell lines, we can find a single good solution which still provides an excellent fit with these constraints. So in conclusion, molecular background can be inferred with sufficient genetic data. Using our model and some machine learning techniques, we can infer molecular background of synthetic data, and preliminary results using experimental data has been extremely promising. Additionally, one of the major challenges when working in theoretical spaces like this is that numerous symmetries exist. And these symmetries can lead to infinite solutions. But here I've detailed some constraints that allow us to overcome these symmetries and find a single good solution. I would like to acknowledge the rest of my lab, the Cancer Evolution Lab, specifically my advisor, Dr. Christopher McFarland, Dr. Mike Helsinski, and my dissertation committee, along with the organizers here. Specifically, Valeria, I believe your name is, who has been working double duty as a presenter and technical support for this session. And I just want to know that I at least really appreciate that. So any questions?
Hi, thanks for the great talk. I think in your presentation, you're assuming for like fitness landscape with a single peak. I wonder whether in the process of like theoretical work or looking at like the real data, whether you've looked at cases where the fitness landscape is a bit more complex, or for example, like a fitness valley.
So it has been on all of our minds throughout the work, the potentiality of multiple peaks, but because there isn't really a basis so far in cancer for what we're expecting, we decided that the best thing to do was start with the simplest plausible landscape, and then if that didn't work, extend it into something that has multiple peaks. Now, technically, Fisher's geometric model doesn't not allow multiple peaks as long as the other peaks are lower than the main peak, because it can be bumpy, but there has to be one optimal solution, not multiple. Thank you for the question.
I have a question.
Okay.
So if this is the Beijing, so how can you measure the fitness? This is the Beijing. Yeah.
Yes.
So, yeah.
So we're using growth rate as a proxy for fitness. There isn't anything that says you actually have to use growth rate. That's just what we've been using. Technically, this can be applied to any measurement that you want. Growth rate is the easiest one because it's very carefully tracked by DepMap. They included unmutated growth rate and mutated growth rate, which is really convenient for us.